Hello, everyone. Thank you for your invitation to the BioKorea Invest Fair. I would like to introduce our main and core solutions. My name is Park Gil-hong. I am the CEO and the representative of the company. As you know, there is no cure for dementia. Among all types of dementia, Alzheimer's disease is very serious when it comes to symptoms, and as a result, many are quite concerned about this type of dementia. This is because of complicated nature of Alzheimer's diseases. This is a CAMP response element and binding protein, and we are focusing on this CREB elements, how to restore them, how to rehabilitate them. And we also have to think about amyloid beta protein deposits, as well as acetylcholine, which is a neural transmission agent. And currently, during clinical trials, the dementia cure is focusing on temporary and short-term improvements of symptoms. That means that you are simply just improving the dose of amyloid and acetylcholine signs, because so as a result, if you take this medicine, then for a few months or a year, you will get better, but that's short-lived. The impact doesn't last for a long time. So what you have to do is that the already damaged neurons or neural features cannot be restored. So at clinical trial number 4, the fourth stage, if you cannot fundamentally change and improve neural transmission or system, then the clinical trial is going to fail. So what we are doing here is to delay the onset of dementia, and what we are trying to do is delayed and modifying dementia symptoms and slowing therapeutics. So we are focusing on a P-tyrosine PKA activator, and it is a protein clinical cure that improves the protein itself, and we're using P-tyrosine dementia prevention. Among dementia, there are many different types of dementia, as I mentioned, and it is related to neural expansion, neural dualization, and usually cyclonic AMP starts the process, and then PKA will be activated, and then it'll activate CRAB, and then BDNF, TRK, AHN, and etc. are going to be impacted, and then as a result, neurons are going to grow and grow in number. It'll split and survive, and it'll also help learning and long-term memory as well. And it also controls the rhythm of sleep, as well as preventing the addiction of a drug or alcohol. But PDE is a enzyme that demolishes CAMP, and currently in global phase 2, 3 clinical trials for novel AD therapeutics are going on. So we would indirectly activate PKA by using P-tyrosine, and that is why we are going through the clinical trials, but we haven't get approvals from the US FDA yet. So all the PDE, which is an indirect methodology, hasn't been able to be launched because of its difficulty, but I would say the direct restoration caused by the use of P-tyrosine is going to show a stronger impact, we believe. And there are many different Peterson types. It starts from A to Z, and as a CRAB controller, we believe in using 1 micromole, and for Peterson AC we use 10 times of those, and it would activate P-CREB according to the dose and the increase of Peterson levels. But what is important here is that astrocyte CREB, there's no impact to it. That means there's almost no side effects on other mechanisms. And next I'm going to talk about whole life cycle activation. As you can see, for neural cells, we give 1 micromole of Peterson derivatives, and then we would give 10 micromoles of AMX, and you can see that pKa, you see this pKa, that is a fermented process, and pKa, pCREB, pTRK, you can see that the AMX control group is showing the similar level of the Peterson derivative level. So this is the structure of Peterson. There is Peterson C and D on the bottom, and there's OHG on the top, and Peterson with OH only will activate PKE as well. And you can see that Aricept donepezil is shown on the right side, and there is a ring over here in the structure, and Peterson has the ring structure as well. So that is why we decided to focus on Peterson for the similar structure compared to donepezil. And what is important is also Peterson does not impact any PDE activation. As a result, CAMP level is not impacted as well. But cyclic AMP is involved in many different physiological mechanisms. So if you move up the level of CAMP, it will cause different side effects. So it shows that we are free from causing side effects. And now I have to think about neurotransmission. You see the pKa structure, there are 2 different domains, A and B, and Peterson is connected to both A and B. That is what we saw. And there are 3 different methodologies. So if you look over here, there is a static connection, and in the middle there is quantum energetic connection, and then there is dynamic connection as well, which is the 3rd one. And if you look at this one, which is a static combination, both C and D, Peterson CD are impacting cyclic AMP, and the strongest combination is witnessed compared to CAMP. So this is CV2. And for quantum energetic connection over here, you can see that it's much stronger connection than CAMP, and also dynamic energetic combination as well. If you look at the analysis, OHB is now showing the feature for improving Peterson and PKC as well. So now we have to look at whether this will actually activate neural transmission too. In general, it does not impact a general neurotransmission, but it's for long-term memory. I can say for sure that it impacted too. Usually memory is taken care of hippocampus multifiber CA1LPT, and this hippocampus multifiber CA1LTP is strengthened by Peterson C and D, and AMF control 10 micromole was given as well. And if you compare the 2 graphs, Peterson C and D are increasing the CREB function in the CA1 region of dorsal hippocampus rescues. And we check and measure long-term quotation and memory, and there is a meaningful growth. But for AMC, there's no statistical proof, whereas for the Patterson C and D, long-term memory was improved for sure. And now I would like to talk about how C3 hydroxylated Pattersons can accelerate neurite outgrowth. You can see the axons and dendrites length have been increased. You can see that the number of neurons have increased. Oh, by the way, Patterson C, we put 1 micromole, and for CAMP, we put 100 micromole. And for Patterson C and D, it is showing that it's increasing meaningfully the number of neurons, and CAMP and CGMP have grown as well. And now I would like to look at whether it improved cognitive abilities of a mouse. So in a water basin, you place a small platform and you would put water, and then mouse is going to kind of rest instead of just diving or swimming. So we checked whether how often the mouth would go to this safe haven from the water. And in this water maze, you can see that normal mouse is going really quickly, and mouse with Alzheimer's disease, it's quite slow. If you put Patterson C, it's moving really, really quickly. And we also want to see how frequently it would go. A normal mouse, they would go really often and rest. And Alzheimer's diseased mouse, they would forget the existence of it and would not go. But if you compare the mouse with Patterson C, they would go and more often than just regular Alzheimer mice as well. And if you focus on Patterson D, the blood-brain barrier penetration level was quite high compared to the control group. We also went through a toxicity test, and we saw that there is no liver cell toxicity. And we also looked at Hep G2 and LDH assay, and there was no measurement of toxicity here as well. So as a result, we chose Patterson D as a candidate for improving Alzheimer's patients and dementia, and it was a strong improver and enhancer of this. So mode of action protein kinase A, KTA, was activated by the use of Patterson D, and some of the study was published at a journal, and they said that there is a strong candidate that could possibly cure dementia as well. There was an editorial research summary that was published by them. And I would like to move on to PKA activator, improving other neural diseases as well. If you look at the table, there is depression, alcohol addiction, insomnia, and ADHD, as well as autism and neural palsy, cerebral palsy, and paraplegia. It could be cured as well. And now I would like to look at what competition is doing at the moment. As you know, there is Eisai and Pfizer who is developing Aricept, and this is a acetylcholinesterase inhibitor, ACHE inhibitor. And all the other competition is focusing on this ACHE, whereas we are focusing on PKE activator, which is a protein kinase A, by using Patterson D, which is first or second line therapy across all dementia types and disease severities. And now we will look at what competition is doing when it comes to clinical studies. Biogen and Eisai's aducanumab is now going through a partial approval from FDA. For us, we are going to pursue clinical trials. Once the first and second clinical goes through, I think we will get expedited approval from FDA because there's no cure for dementia patients at the moment, and then we're thinking of going through IPO as well. So besides dementia cures or treatments, we do have other pipelines. We have arthritis prevention mechanisms, and we're targeting low molecular methodology, and we are also studying on hair loss prevention, and there's also general antibiotics as well, because for our antibiotics, it's RNA polymer inhibitor, and this could also work against COVID-19 pandemic or virus. It does. The general multi-use antibiotics could kill COVID virus and make sure that antibodies are enhanced, and we check this by putting a lot of investment onto this specific project and saw the efficacy. And now let me move on to global strategies. So we are going to export Miracle Morning, which is a cognitive capability approval, and we got an approval by US FDA. We are going to export this product to other countries. And for licensing out portability, I would say is that currently there's no fundamental cure for dementia, and at the same time, dementia patients, dementia cures would cost about $1 trillion to get an approval from the US FDA. So I believe that if our clinical trials go through and clinical phase 1 goes through successfully, licensing out would be better for by targeting global pharmaceutical companies so they can use our technology for production. And I believe that there will be a phase of a quantum jump from our company. We are located at the Korea University Medical Center. We are a VC affiliated research center as well. And we also are targeting many types of chronic and rare diseases. We own and registered a total of 18 different patents in Korea and outside of Korea. And we are a subsidiary company of the Korean Medical Group, and we're affiliated with other public organizations and university hospitals and foundations as well. These are the list of our partners if you're interested. This is the organization chart of our company, and this is my profile and history and career. And these are some of the core people in our startup team as well. So currently, the dementia treatment is being developed by Professor Jae-young Ko at Seoul Asan Medical Center, and he is a renowned specialist for dementia, and that's why I would like to introduce his leadership in our project. Thank you very much for your attention. Thank you.